The world of pharma regulations is constantly evolving, presenting new challenges and opportunities. In this ever-changing environment, continuous learning is key. Therefore, Express Pharma is launching Regulatory Watch, a series of masterclasses led by experts and thought leaders to empower you with the knowledge and insights needed to navigate the ever-changing regulatory landscape successfully. The second masterclass in this series is by Mr Masihuddin Jaigirdar on product and process-related regulatory issues in the submitted ANDAs. It will be divided into 3 sessions. The first session highlighted the critical areas to be focused on for quality submissions, and the second session covered drug product and manufacturing. This session, the third and concluding session, will focus on common deficiencies during scale-up. Mr. Masihuddin Jaigirdar is a pharmaceutics expert specializing in advising companies on compliance with US FDA regulations. Before retiring, he was a senior quality reviewer with the US FDA's Office of Pharmaceutical Manufacturing Assessment. This position required ensuring that the drug product and process quality was in line with principles of quality by design QBD for NDAs, ANDAs, about chemistry, manufacturing process, controls, stability, and environmental impact. He received a certificate of appreciation from the CDER, FDA in October 2020 for his dedication to the FDA through his 10 years of exemplary service. His industry experience of over 35 years spans the RNMP, D generic divisions of Actavis, former Watson Pharmaceuticals, Hochst Marion and Russell, Marion Merrill Doe, former Chelsea, Mylan Pharmaceuticals, and Astra Development AB, former Kipico Process Technique.
Now about the scale-up. Proposed process parameter for commercial scale not supported by established scale-up principle. Here is an example. Farm has proposed for the commercial batch in the 2000 liter AC blender, having same blending and lubricating times of 10 minutes and 5 minutes respectively, as that of their 750 liter AC blender used for the exhibit batch. This is very unusual, and with my 35 years in the industry, I can tell you this is not possible. Though for diffusion type mixer, moving to a larger blender, the same number of revolution will be required. However, the blend time would increase due to the fact the larger the blender, the slower the vessel rotates. Remember, if the larger blender is at the same as the smaller blender, it will go out of your room of production because it will have, by law of gravity, it will have a high momentum. So this is not possible. So I have asked, please justify your rationale for having same mixing time between these two blenders. Also, please provide the rotational speed, RPM, for the 2000 AC blender. Again, this is impossible, so farm has to see why between the 750 liter and 2000 liter, they are proposing the same mixing time, and it is just not possible. Another example is farm has used 30-station fatty tablet press for the compression of exhibit batch tablets and proposed to use a 49-station fatty tablet press for the commercial batch. So we have requested to provide comparative press speed to maintain their same dwell time for scale-up perspective. One has to remember, it is important for the tablet compression, comparative dwell time. It doesn't matter what type of or what number of station you are using, the dwell time for between these the tablet press used for the exhibit batch and the commercial batch has to be same. that is more important than the speed of the press. So by adjusting the speed of the press, you have to have comparative real time. I have an education in pharmaceutics and many years of experience as product development scientist. And later I have worked for the FDA as its senior CMC rep. And my total professional exposure is about 50 years. What I have observed and found that the technology transfer and scale-up strategy from R&D laboratory batch to exhibit batch and then to the commercial batch is not fully understood and well taken into account. And I am talking about these ANDAs coming from abroad. It is mostly likely that many applicants are using locally fabricated equipment. Their manufacturer do not understand the basic principle of scale-up. Please concentrate in this area, which most of the time involves law of physics and mechanics. This is not chemistry. So for the equipment operating principle, including T-speed and the scale-up based on mathematics. Common deficiencies manufacturing process description. Your manufacturing process has many subjective control which are operator dependent, difficult to control and lead change of variability in the process and end result. Please employ more qualitative control, I would say quantitative control for the following. The amount of granulating solution and its rate of addition. So granulating solution, you have to know how much you are adding and the rate, that how many grams per minute you are adding, of how many liters per minute you are adding, or per hour you are adding. So it should not be just I'm putting the granulating solution randomly. So rate of addition and amount has to be determined. The amount of wet mass and its thickness on a tray. Say if you are doing in an olden days, still using tray drying, so you must know that how many trays you are using, and there should be a same amount of wet mass and thickness. There should not be uneven distribution. It should be evenly distributed, so the thickness on the tray should be same. And the drying time, by establishing a drying curve to achieve dried granules having enough strength for comminuting, which is sifting and milling. Also establish granulation endpoint by either measuring electric power consumption amperage or impeller torque, which is resistance in terms of ohms of the high shear mixer granulator. For the sifting and milling step, you have emphasized the requirement of passing all the dried granules through 30 strain. Please explain this requirement from critical parameter perspective and also include test and specification for particle size distribution of the final blend. Common deficiencies for the microbiological control. Farms microbial limits acceptance criteria are less stringent than those provided in the USP chapter 1111. Microbial examination of non-sterile product acceptance criteria for pharmaceutical preparation and substance for pharmaceutical uses. I'm not going through this. I'm just going through what was the deficiency I have given. The proposed microbiology innovation acceptance criteria are not consistent with the recommendation for non-aqueous oral preparations in USP 1111 provide a justification for the proposed exception criteria. 23P24 Container Closure System. Examples of common deficiency in container closure system. What specific container closure attributes are necessary to ensure drug product performance? So I have given the following deficiencies. Please provide USB testing data for your container closure system demonstrating compliance with current USP 671 for single unit container blister or tablets. Your stability studies for the drug product package in both proposed container closure system at 3 months accelerated conditions show a trend of loss in potency. Please explain this trending. This is about Gudufar impact. So this can be maybe a measure if there is a lack of or inadequate critical in-process related controls such that the firm cannot demonstrate it has the ability to consistently manufacture a high quality product. These are the common abbreviation used and I have given and you can go through.
Thank you for watching session 3 of Masihuddin Jaigirdar's Masterclass, focusing on common deficiencies during scale-up. Don't miss session 1 and session 2 to complete the Masterclass.